Hello, everyone. All right, so I've been listening intently all day, and my mind's just kind of a blur, as yours too. I'll tell you about Ajax. You know what I think I'll do is, I'm glad I put some basic principles in aging in here, because maybe it's kind of good to step back and kind of look at what we've been listening to. We've been hearing about transthyretin and amyloid and stem cells and electroporation, all kinds of things. So maybe it'd be kind of good, I'll give you a little bit of a background and then tell you about what this company, Ajax Therapeutics, is doing. Ajax is a subsidiary of a public company called Biotime. BTX is the ticker, and so I'm required, by law not to read this, but to remind you that we have filings with the Securities Exchange Commission. I'll also say that I wanted to thank Kazoo Technology Capital and Jim Mellon, who obviously are all here, who are investors in this new venture. I thank them for vision. There's so much vision represented here today, and for what I'm going to tell you about. I'll also thank a scientist who's no longer with us, but about 130 years ago, August Weismann, a brilliant scientist. I think he's one of the brightest biologists in history, a German naturalist, pointed out some very basic biology, and if he was here today, he'd probably start with this slide. So it's a fundamental fact of nature. It's like the birds and the bees, but one that's good to come back and think about for a second. I've been working on aging for a long time, maybe 40 years or something. Well, I've been working on aging myself since I was born, and before, but the biology of aging has been of interest of mine. And this principle of biology has always been my starting point. So what it's reminding us is that you and I are made from a lineage of cells that have been proliferating for billions of years. Unlike our ancestors, we have dead ancestors, but our cells have no dead ancestors. Cells that made us have been seeing free radical damage and all this kind of extrinsic wear and tear, and have perpetuated the species and made you and me. The difference is, of course, the cells that spin off from that, this disposable soma that August Weismann always talked about, are very different. And so the starting point of everything I'm going to talk to you about today is that dichotomy, germline soma dichotomy. Forgive the French, but what the hell is wrong with our somatic cells? What's going on? And I'm going to talk about reprogramming. So Weismann, this is him on the right, is famous for this quote. He wrote an essay in 1881 called The Duration of Life, and this is a quote from his essay. And he said, "Death takes place because a worn-out tissue cannot forever renew itself, and because of capacity by means of cell division is not everlasting, but finite." You notice I'm going to highlight two things here. There is a conjunction here, and the first thing he said, that a worn-out tissue cannot forever renew itself, and the capacity for cell division is finite, not everlasting. He meant, of course, in the soma, right? In you and me, not in the germline, of course. Okay, point number two. I called this little talk the reprogrammability of aging. Well, to reprogram something implies that there's a program, right? And aging researchers for years resisted that idea. They said, we don't age from a program. Nature never selected for a genetic program of aging. And the reason is that aging is normally thought of as something that happens after reproduction, and so evolution only cared about selecting for traits involved in reproduction and before, not at what happens to you when you're 80 or 100 years old. then along came this man, George Williams, who's an evolutionary biologist, and he proposed something which is kind of stuck in the aging community, which is this idea of antagonistic pleiotropy. So in his essay, Williams proposed that there is a program of aging in this sense. There can be genes that are pleiotropic in their effects. So the sequence of that gene can have one effect late in life that's different from the effect early in life. And nature selects for something that benefits you early on, but that same inherited trait can be deleterious later on. And that's the antagonism of pleiotropy. So there is, according to this model, a selected genetic model of aging. So if you take all this together, Weissman's idea and Williams' idea, what I could argue we should be focused on is molecular changes that are involved in this germline soma dichotomy. The antagonism could easily be tumor suppression, right? So whatever these traits are, they're trying to prevent cancer early in life, but the benefit or the antagonism would be that they cause aging. And so the prediction, if all that's true, is that the things we're looking for should be changing sometime early in life and then reappearing in the context of cancer. That's sort of a test of whether this hypothesis is true and or whether these pathways play a role in aging. All right, so you all know about telomerase. So years ago, Alexei Lovnikov proposed this wild and crazy idea that telomeres shorten in somatic cells and that there was an enzyme that made telomeres and they made cells immortal, and it was turned off, causing cells to age in our body, somatic cells, but then reappears in cancer. I'm going to quickly review some of that, and then I'm going to talk about another idea which we're developing at Ajax, which is another thing that occurs that meets these criteria, is regeneration, okay? So the idea is that regeneration, that there's genes controlling regeneration that are turned off after the body is formed, and that plays out as aging later in life, and it reemerges in cancer. So cancer can be seen as immortal regeneration out of control. All right, so that's what I'm going to be talking to you about. Well, first off, when do these events occur temporally? I don't have any reason to believe that these two phenomena, and by the way, I'm sticking my neck out here. I'm suggesting to you here that there are two mechanisms that are fundamentally important in aging and may be the majority of aging. One is the repression of telomerase in the soma, and the other is the repression of regeneration. That's the thesis, the theme of what I'm telling you about today. I don't have any reason to believe that they're really linked, except that they both evolved for the same purpose, through breast cancer. Telomerase turns off in the soma very early. I mean, when we study embryonic stem cell differentiation, I mean, expression of telomerase vaporizes within days in everything we've ever looked at. Regeneration, we know, turns off once the body's formed. So I'm showing you here 8 weeks of human development. That's when you transition into a fetus. So that's called the embryonic fetal transition. And what's interesting about that transition is the body's formed, and then about contemporaneously with that, the body's sort of saying, we've made everything. We've done this organogenesis. We could turn all the ability to do that off. It'd help prevent cancer. But then, of course, you can't grow a kidney again. You can't we'll replace it with scarring. We'll allow the body to scar to do a healing, but we won't allow the body to regenerate. So that's the concept. All right, so if we're thinking about taking the immortality of the germline and transferring it into the body somehow, how can we do that? Well, one idea was, of course, telomerase. And so Len Hayflick showed that somatic cells age, all human somatic cells age when cultured in a dish, except for embryonic stem cells, which sort of mimic the germline. They are a very primitive cell that can even make germline cells. and so cells lose telomeres in the soma, as we probably all know. The remarkable thing was when we cloned telomerase at Geron and dropped it into, okay, I just got to tell this story. I told someone over lunch, some of you may have heard me say this before. Len Hayflick, for years, said we'd never understand the aging of human cells in the dish, in vitro. There are over 300 changes. It's very complex. He wrote papers on this. You can look it up. So when Geron, when we had the telomerase gene cloned, we actually had his cells from his leg in the dish. He gave us some of his skin. And the first human cells ever to be immortalized with telomerase were his cells. And I had the fun chance to tell him that one gene had immortalized his cells, despite the apparent complexity of all these hundreds of changes that are occurring. I'm just telling you that because there's no reason to be certain that the aging of the entire organism is not parallel to that, where we think, oh, there's thousands of changes going on. There could be some central mechanisms. Could be. Anyway, what really got us excited about telomerase was it fit the model. Over 90% of human cancer cell types immortalize and turn on telomerase. And if they don't use telomerase, they maintain telomeres by alternative mechanisms. That fits this model, right? Antagonistic pleiotropy. Well, that then led to this, you had heard Jean Lauren talk about embryonic stem cells. That led to this idea, could we capture the germline in the dish, embryonic stem cells, and use them? They would be predicted to have telomerase and be immortal. That would allow you to expand these cells indefinitely and have an industrial platform to manufacture young cells of any kind to replace dysfunctional cells and tissues, like in Parkinson's or many other applications, of course. And these are the cells that were isolated back in the late 1990s. And there are many different cell types, as we've heard about, that you can make from these cells. So at Biotime, we're developing some of these cells, and our subsidiary Asterius, some others, and in Ajax, we're working on additional ones. Another thing that was really profound about this was the transfer of a somatic cell into an egg cell can reprogram cells. And back in the year 2000, we took cells to the Hayflick limit, bovine cells in this case, and showed that nuclear transfer could reset the telomere clock even longer than it was originally. So you'll see this curve that goes out beyond the original cell lifespan of cells. Those were derived from senescent cells, and we reprogrammed them to even longer telomere length than they had originally. And these animals you see cloned on the right were all cloned from senescent cells, full of lipofuscin, you know, full of wear and tear, and they had normal, if not longer than normal, lifespans. This idea that cloned animals are prematurely old is just nonsense. And of course, now this can be done using transcriptional reprogramming. So on the left, you see telomere length at the beginning of life, embryonic stem cells, cells grown to senescence, and then the resetting of telomere length over time. And then the lifespan graphs, you can see how we can reset cell lifespan based on telomere length reprogramming. And then on the right, I'm showing you the Horvath clock. This is his published data on showing that even this methylation clock of aging gets reset and reprogramming. So if we turned human reprogrammed cells into a baby, which we could do, all the indications are, as through cloning, that that would be a baby that would be born young and would live a normal lifespan. That's pretty remarkable, really, if you think about it. We could now make young heart muscle for any of us forever, or any other replacement cell type of the human body. Who would have thunk a few years ago? So are there practical applications? Of course. We at Biotime, the parent company, are making young retinal pigment epithelial cells. I think this is a sort of classic example of heterochronic, meaning young cells in old people, transplantation for an important age-related disease. I won't go into all the detail, but you guys know that I think that the aging of this layer of cells in the back of the retina trigger both the wet and the dry form of macular degeneration, a really serious problem in aging. And we're targeting the dry form, of which there's currently no known therapy, no approved therapy. And the wet form of the disease is treated with existing drugs and sell $7 to $8 billion a year of product. So the dry form is a really significant unmet need. We're in phase 2 clinical trials with these cells in Israel and in the United States. One cell type that we're developing in Ajax are brown adipocytes. So on the left you can see there's an age-related loss of these cells in you and I. What's important about these cells is that, which are just recently appreciated in the human species, is that they are sort of the opposite of white fat that stores calories. They burn calories. They were recognized early on because of their dramatic ability to pull glucose out of the circulation. That's how they were imaged and recognized in humans. And they secrete at least 5 known cardioprotective agents, adipokines and other molecules. And so the thought is that they, when they're ablated in an animal, you get obese, diabetic animals, and when you restore brown fat, you see weight loss and insulin sensitivity return. We can make them in a very effective manner, essentially 100% pure, using a technology we invented called PureStem. This is UCP1, it's a marker of those cells on the right. On the left you can see fetal source cells which are not pure. And so we can make them in vitro superior to other means of sourcing the cells. This is of course an important application because up to half of Americans are anticipated to be pre-diabetic, not just Americans, around the world. Everyone thinks of Americans as obese. Why is that? We do have an obesity problem there, and other places too. We're making young vascular progenitors. We can make vascular endothelium using our PureStem technology. And if you just look at that little flow sorting insert there, you can see 100% pure embryonic vascular endothelium and smooth muscle progenitors. That's another program in Ajax. And of course, cardiovascular disease is an enormous market. All right, well, the last thing I want to talk to you about is, remember I said, the maybe 2 things involved in aging? It's what I want to propose. What's the other one? Regeneration. In this essay of Williams, in blue is a quote from, I think, the first page of his essay. Indeed, it's remarkable that a seemingly miraculous feat of morphogenesis of a complex metazoan, or you and me, should be unable to perform the much simpler task of simply maintaining that which has already been made. That's true, isn't it? When I read that, it just, bam, that's right. That's really weird. Well, let's look at this evolutionary tree. Many single-celled animals are immortal, tetrahymena, for instance. Then you move up this evolutionary tree, Hydra, Planaria are immortal. But what I'm going to propose to you is that there's this barrier, the Weissman barrier, the red line, and some animals have crossed that line. And we cross it during development at the embryonic fetal transition. And let me give you some examples. Hydra don't age. Look at the survivorship curve of Hydra. I think you guys know this maybe. You can lob them in pieces and they grow back, and there's no known aging in Hydra. Planaria are another example. Early researchers in Planaria, relatively complex metazoan by the way, with a nervous system and eyes, said that they're immortal under the edge of the knife. Watch the eyes appear in that tail. Isn't that amazing? Just awesome. No known aging. They have telomerase, and they have complete regenerative potential within reason. I mean, if you cut them into 1 million pieces, they won't grow back. These salamanders you've all seen regrow are stuck in an embryonic state, so they regenerate. Cut the leg off, it just grows back. They were a normal animal. If you fully differentiate them with thyroid hormone, which they're missing, then it represses this. But this is something they have in the embryonic state. Humans have it too, right? Embryonic skin in humans regenerates, for instance. So does the central nervous system. So does the heart. Well, in collaboration with in silico medicine, Alex spoke earlier, and some other work we've been doing, we've been mapping genes that change since embryonic stem cells are on this side of the Weissman barrier, and we have many cells that are on the other side. We did, genomic analysis. Here's an example of one gene on the right. Here at the left, you can see adult-derived cells and embryonic on the left. This gene turns on during adult development. Top right, you can see it come up during age, and it's turned off in cancer. Look at the bottom right. It fits this model, but we have other genes as well. So what we're working on in AGEX is to induce tissue regeneration. It's kind of like this thing you heard about partial reprogramming. I don't like the term partial reprogramming because we all know if you do IPS reprogramming, the cells are really confused for a while. We're just trying to target the induction of regeneration. IPS reprogramming resets this regenerative state, but we just want to induce the regenerative state and induce telomerase to make us into flatworms, so to speak. Can we make sense? I started out talking about all the stuff you've been hearing about. All these different things we hear about aging. A lot of pathways. You notice all these arrows when people talk about molecular pathways of aging, they all point and they just say aging. But how do they influence aging? It's always a question mark. What I'm suggesting is all these pathways are involved in growth and differentiation, and the way they affect aging is by inducing tissue regeneration. Okay? mTOR, growth hormone. I'm suggesting they all allow the body to regenerate, and that is how they influence aging. It's a proposal. During Q&A, you can tell me I'm wrong. So another little thing, it's tempting to speculate that since this is very attractive to me, if a tissue can regenerate, then the cells, if they have genotoxic damage, have P16, they have DNA damage, the body can say, get rid of them. We'll just make more. If regeneration's turned off, like in the myocardium, and you have genotoxic damage, nature may have said, hey, let's block apoptosis. We don't want to lose that heart muscle cell, we need it. You see what I'm saying? So what we think is that by inducing tissue regeneration, we can allow a natural senolysis. Those cells will be thrown away, and then new cells will be grown to replace them. It's a very attractive model, at least theoretically. These degenerative diseases, of course, are enormous markets, and that's why AGEX is focused on the regenerative aspects of all this technology. I'll summarize with this quote. A famous person studying regeneration opened his essay saying, if there's no regeneration, there would be no life. If everything regenerated, there'd be no death. Something to think about. Now, I'll quit with that and take Q&A.
Yes, thank you very much.